So we will start by the presentation of Aki Sinkkonen. Aki is a researcher at the Resources Institute Finland, focusing on the environmental biodiversity and its role in human health. And he's particularly interested in how the interactions between the ecosystem and the exposome affect the development of IMDs. So Aki, please, the floor is yours.
So I'm Aki Sinkkonen, principal scientist at the Natural Resources Institute Finland, and I'm going to talk about the role of environmental biodiversity in developing IMDs, immune-mediated diseases. I have studied 10 years urban biodiversity and done urban biodiversity interventions, many kinds of interventions. And I hope I'm a specialist in the interlinkages between the environment and human microbiome. And the question for today is, why do daily contacts with environmental microbiome matter? And here are two photos. The history photo, it's from 1940. Then we had dirt, and dirt is rich in environmental microbiota. It was everywhere in our life, but nowadays, when children grow up, there's hardly any dirt. Several comparative studies have shown, or actually many of them, that poor human microbiota is linked to IMDs, particularly atopy and type 1 diabetes, and in some cases also asthma and allergies. And there's a kind of consensus that our lifestyle and inorganic surface that doesn't let water go through, so they prevent daily contacts and the beneficial exposure to environmental microbiome, to organic soil and to plants. And last year we published an estimate that in Finland, where the population is only 5.5 million people, so the causes of asthma and type 2 diabetes and depression due to lack of nature contacts. So it's between 140 and 340 million euros. So if we extrapolate this to the EU level, so it's much more, 50 times more, or even more. In HEDIMED, we have asked if environmental microbiodiversity reduces the risk of immune-mediated diseases. We have done it in two ways. We have done satellite database studies where we have searched for correlations between IMD risk and exposure to environmental microbiota. In those studies, we used land cover type as an indicator of exposure to environmental microbiota. Then the more specific type of studies type, they are biodiversity interventions. We have added biodiversity, visible biodiversity, to everyday living environment, daily life of urbanites. You can see that the photos, it's a change of a standard daycare yard to a rewilded daycare yard. And we have studied potential effects on immune modulation and potential effects on disease incidence in short-term and long-term intervention trials. This slide is about the land cover studies. Our conclusion is that wild and semi-wild areas, they are much better than manicured lawns or sealed surface. In those wild and semi-wild areas, like agricultural areas, the risk of atopic sensitization, it was smaller in a study made by our Estonian partners, and then risk of type 1 diabetes delayed if the birth address of children contained plenty of agricultural land cover. This study was led by the Tampere group in HEDIMED. You can see how it was done. The different radiuses around the birth address in those both studies, and then we checked which land covers are within those radiuses. Here's one of the intervention trials. This is a short-term intervention trials. In this study, we recruited daycare children, normal urbanites between the age of 3 and 5. And we let them play in sandboxes that we built by ourselves. But we filled the sandboxes either with peat, which is low in microbial diversity, or extremely rich soil, so microbially rich soil. And then we let the children play there for 2 weeks, 2 times 20 minutes per day, so from Monday to Friday. And we measured the commensal microbiota and immune response before and after the intervention period. So this was a double-blinded randomized intervention trial at the daycare yards. You can see how the children were supervised for 2 weeks. This is an example about it. And we also observed shifts in microbiota and immune modulation in the intervention group, where children played in sand mixed with microbiologically rich soil, not when the sand, there was only peat, microbiologically poor peat in the sandbox. And we also saw how immune regulation enhanced. And this is a graph about it. You can see that in the intervention group, the change was positive and higher. This is a change of an immunomodulatory cytokine, so an immune marker measured from blood, that is interleukin-10. But in the placebo group, there was no increase during the intervention period. This is an indication that immune regulation enhanced due to this intervention. This is the film about our long-term intervention trial. In this trial, our target is to recruit 350 children. This is also a double-blinded trial. It's placebo-controlled, so the parents, they use lotion every day. It is either microbiologically rich lotion or it is placebo. And they also use hats, so they wear hats to their children, and the hats also contain microbiologically rich material or placebo, and they also use sheets that have the same thing, either placebo or microbiologically rich material. And this way, babies between the age of 2 and 12 months, they are exposed to, or half of them are exposed to biodiversity, and the other half receive the normal biodiversity of urban environment. And then we follow the children until the age of 36 months, so 3 years, and we measure IgE sensitization, so allergic sensitization. We follow it from the age of 2 months to the age of 3 years. This is a double-blinded trial. This started before the COVID pandemic, and unfortunately, due to recruitment breaks caused by COVID conditions, so this is still an ongoing study, and therefore we do not have the results. We know that this is a safe trial. We have recruited almost 200 children so far, and there are no side effects, but we do not, unfortunately, know the results. We will study many things from the children, not only microbiota and IgE sensitization, but we will use all the modern available techniques to see how this intervention affects the health outcomes of urban children in Finland. And there's an important thing. The children that are recruited, the families that are recruited, the children are high-risk children, so both of their parents have atopy, which means that we can say that a half of the children would get atopy without the intervention. So this is the policy relevance of our HEDIMED findings. We recommend being in contact with natural and semi-natural areas, rewilding of urban areas. This means that we add visible biodiversity. At the same time, we add microbiological biodiversity, because environmental microbes live in visible objects like organic soil, dirt, and plant. And rewilding means this adding biodiversity to urban living environment. So it's important. All the levels from local municipalities to governmental and EU level regulators, so they should be aware of our findings. And the next steps, we recommend national and local action programs, also at the EU level. What we need, this is almost the same as the previous slide, so daily contact with the rich and abundant microbiota, particularly in early childhood, rewilding of urban environments, if it's not possible to rewild the whole city, so at least those places where vulnerable groups like children where they spend their time. And unsealing, so taking off the non-permeable surface. What's next? We need long-term biodiversity interventions, preferably double-blinded trials. The target is to see if they affect disease incidence. Those can be indoor and outdoor rewilding trials. Outdoor rewilding trials, they seldomly can be double-blinded, but they are also very important. We need studies in several climate zones, not only in Finland, in boreal or hemi-boreal zone, but also in Mediterranean and broadleaf areas. And collaboration between medical scientists and environmental scientists, it's a unique character in HEDIMED. And of course, finalizing and publishing ongoing long-term trials delayed by COVID-19. Thank you so much.